Edward Taylor once said, "The science of today is the technology of tomorrow." Hello, my name is Marco Muñoz, and today I'm going to talk about eukaryotic gene modification by a CRISPR-Cas method. One way scientists have been able to modify genes, especially in eukaryotic cells, is via the CRISPR-Cas method. CRISP stands for Clustered Regularly Interspaced Short Palindromic Repeats. When CRISPR is combined with appropriate enzymes and molecules within the genome, it can create a complex tool that serves for editing, copy, and paste DNA segments. This did not happen from one day to the next. In fact, it took 20 years of research and 12 cities from 9 different countries to fully understand CRISPR and its mechanism. And it wasn't until 2012 the scientists were able to edit genomes in mammal cells. Gene modification is a powerful tool that brings many benefits. However, there is opposition regarding CRISPR despite the many great possibilities CRISPR can provide. So what is CRISPR? CRISPR originates from bacteria and archaea cells located in their genome at a specific loci, and it is known to be an immune adaptation developed as a response against invaders such as viruses. CRISPR was first discovered by Francisco Mojica in 1989 while experimenting with archaeal microbes. Currently, there are many different types of CRISPR based on the complementary elements. However, all of them resemble similar structure and functionality. The simplest of all have been the basis for genome editing purposes, CRISPR-Cas9. CRISPR in a given locus consists in 3 key players: a CRISPR array. This is a sequence of unique DNA separated by spacers, and these spacers are small segments of foreign DNA, and they are somewhat palindromic, which means it could be read the same as backward and forwards. Next, there are 4 protein coding genes attached: Cas1, Cas2, CNS2, and Cas9. And at the end, there is tracRNA, a fundamental enzyme that associates with Cas9 protein once it is transcribed. How does CRISPR system work? CRISPR follows 3 stages: acquisition, cRNA biogenesis, and interference. Acquisition begins with the recognition of foreign DNA. Cas1, Cas2, and CNS2 identify these segments, capture it, and insert them into CRISPR array. CRISPR array could be constituted by 50 units of these called spacers. The cRNA biogenesis stage involves the transcription of the CRISPR array and transcription of the tracRNA. These 2 transcribed RNAs fuse with the system of Cas9 and RNase enzyme. The combination results in a trans-activating cRNA. In the last stage, interference, cRNA combines with Cas9 enzyme and forms a complex CRISPR-Cas9 endonuclease enzyme that serves to break a double-strand foreign DNA whenever these are present. However, CRISPR-Cas9 just don't work by itself. It must be guided prior cleavage. Another key elements: gRNA enzyme and PAM. PAM is a 2 to 6 base pair DNA sequence. The gRNA directs CRISPR-Cas9, and PAM tells CRISPR where to start cutting. In 2008, the idea of having CRISPR introduced to a human genome was a big if. Mammal cells are more complex and have very different internal environments than other microorganisms do. However, by 2012, Feng Zhang, a Harvard student, became so interested in CRISPR that decided to dedicate his time to develop an engineered version of CRISPR-Cas9 stable enough for the human cells. Zhang experimented with Streptococcus thermophilus and Streptococcus pyogenesis bacteria, and by the end of 2012 successfully accomplished this task. Later, scientists started testing a model version of the created CRISPR-Cas9 enzyme in lab rats, and as a result, it was found that multiple genes could be edited simultaneously by programming the CRISPR array with the spacers matching the given sequence. What could be accomplished by using CRISPR-Cas gene editing? In recent years, it was discovered that CRISPR system can serve as a gene drive for the control of insect vectors of diseases. Malaria is an example of a disease transmitted by a mosquito around the world, with Africa being the most affected. Malaria kills thousands of persons each day, and more than 50% are children under the age of 5 years. The worst part is there is no vaccine that can prevent this disease. A recent article titled CRISPR-Cas9 Genes Drives System Targeting Female Reproduction in the Malaria Mosquito Vector Anopheles gambia. A group of scientists inspired by homing endonuclease genes decided to test CRISPR-Cas9 endonuclease enzyme with the objective of reducing the reproductive capability of the insect vector. The major challenge encountered during the process was the somatic disruption. To avoid this issue, scientists developed CRISPR-based gene drive system designed to home, called CRISPR-H. CRISPR-H was introduced in the germline during the process of gamete formation. As a result, each integration event generated a CRISPR-H allele encoding a Cas gRNA endonuclease designed to target the corresponding site on the chromosome. In the visual screening performed to the offspring, it was noticed a supermendelian inheritance of the CRISPR-H allele with the rates of 94 up to 100%. CRISPR-Cas9 gene drive was successfully passed on to the next generation. There was no obvious decrease in homing performance in across generations over time based on the examination. This could be used to suppress a mosquito population to levels that do not support malaria transmission. Therefore, this development could save thousands of lives in the future perspective. Not only malaria disease could be controlled, but this might bring other ways to develop more solutions to combat dangerous and mortal diseases, including cancer. But what could go wrong? These forced changes in nature can have a negative impact on the ecosystem if the whole mosquito population is eradicated. If these species are gone, then it could affect the whole food chain. Because mosquitoes are a good source of nutrients by other living organisms and contribute in pollinating flowers too. Another thing that could affect us is the replacement of mosquitoes by other species that can develop other serious diseases and bring chaos to the world. In this video, I explain how CRISPR was discovered and how its mechanism works. It was seen in detail about CRISPR components and the stages the cell has to endure in order to activate these enzymes. A given example using an article illustrated the other functions of CRISPR and the pros and cons of the given example. Gene editing by CRISPR-Cas9 method has opened a door for many scientists for a better or for the worse.